globally, there's probably millions of species of bacteria, yet only very little bit of morphological variation. And most of that variation probably happens in the cyanobacteria, which we'll get to. Now, so cyanobacteria produce oxygen, they're photosynthetic, and they're the organisms that oxidize the planet, starting probably about 3.5 billion years ago. And by 2.5 billion years ago, they made it possible for multicellular organisms to do well because the oxygen makes for more efficient respiration. They also fix nitrogen with heterocysts. What fixed nitrogen means is that they can use atmospheric N2 gas as a source of nitrogen. And that's different from essentially all other eukaryotic organisms that have to have their nitrogen in the form of either organic nitrogen or inorganic forms of nitrogen, such as nitrate or ammonia. Okay, so it gives them a big competitive advantage when there's not a lot of nitrogen around the environment. They also are able to float. So when we were at the Kimball Pond, there were the scums, especially when we first got there, were up right on the surface. And that's because they have these protein vesicles that they synthesize, and they have a hydrophobic inner part, so the water won't get in there, the water vapor won't work its way in there, and a hydrophilic outer part. So they basically make these vesicles or bubbles inside of their cells that cause their density to be less than the density of water, so they float to the top. These are what give, when you see the cyanobacteria in the microscope, they look blue-green because of their pigments, right? They have the special pigments that absorb in the green region. And they also look sort of grainy or sparkly, and that's because of those gas vesicles. They give it that kind of grainy texture. So this gives them a big competitive advantage. Why do you think being able to float on the surface, and they're common in eutrophic lakes or ponds, why do you think being able to float on the surface gives them a big competitive advantage? Yeah, they shade everybody else out, so it's perfect opportunity for them to win. Related to the goods and services that people are interested in fresh waters, they're problematic because they cause taste and odor problems in drinking waters. And we're lucky here in Manhattan because our drinking water comes from groundwater, so we don't have that issue. The taste and odor, any taste and odors that are in our water are probably not related to cyanobacteria. But many communities use surface water and they have cyanobacterial blooms. They create a compound called geosmin, G-E-O-S-M-I-N, geosmin. And that compound has sort of a musty or a moldy flavor to it. If you go to Wichita, Dallas, just all kinds of cities around the country, in late summer, and you get in the shower and you turn on the shower, there's this sort of musty smell that comes out at you, and that's this geosmin kind of being volatilized as you take your shower. In addition to that, many species, probably all species, produce some sort of toxins, but some of the toxins are pretty bad, and I'll talk about those in a second. And then we talked just already briefly about these unique light harvesting properties that make them look blue-green or cyano color. It also makes them able to compete well in low light situations. So again, we're talking about they do well in eutrophic conditions. Is light attenuation high or low in eutrophic conditions? Is light attenuated rapidly, or is it transmitted more effectively when it's eutrophic?
Attenuates faster in eutrophic.
Right, it disappears faster with depth in eutrophic. So when we were at the eutrophic pond, we were losing the second depth disk in centimeters. When we were at the mesotrophic eutrophic lake, we were more like in the order of meters to 2 meters. Right. So if they are able to deal well with low light, then that gives them another advantage, because if the wind mixes them down at all, right, they're going to be in an area that they can't photosynthesize very well. And if they have these extra pigments, it's going to make it that much easier for them to do well. So I mentioned that cyanobacteria have quite a few different morphologies, and they can be single cellular, or a few cells, or multicellular assemblages. They have specialized cells, and I talked about heterocysts, and we'll get more of those when we talk about the nitrogen cycle. But this cell right here is a site of nitrogen fixation. There's another one right here, right here. Some species have them, some don't. These long skinny ones are heterocysts. They also, some species have resting cells called akinines. And you can even get a sense of the sort of granular appearance they get from their gas vesicles. So these can be either planktonic and floating like these are, or benthic and attached like these are. Right, so they have a variety of different ways that they can do things. One of the things that has come about is that it does appear that their morphology really is related in a general sense to their taxonomy. So the molecular analysis confirms that you can actually use visual morphology to do a relatively good job of distinguishing species of cyanobacteria. So cyanobacteria produce at least two major classes of toxins, hepatotoxins and neurotoxins. Where do the hepatotoxins act? Liver, right? Hepatic, liver. So they either can get you in the liver or they can get your nervous system. Both of these are the neurotoxins are extraordinarily toxic if you get them in high enough concentrations. Also called very rapid death factor. And if you take a toxic bloom and basically break the cells and put it into a mouse, it's just gone. Right? So that's one of the quick tests for if it's a toxic cyanobacterial bloom. The hepatotoxins damage the liver because they inhibit phosphatases, and basically all organisms have phosphatases, right? You have to have phosphatases to do things like convert between ADP and ATP, right? To take ATP to ADP. So all your basic energy needs are met because of phosphatases, and there's all kinds of other phosphatases in the cell. So both of these are actually very broad spectrum concentrations, very broad spectrum, and they evolved as protection against grazing. So why do you think they're so broad spectrum? What do you think it is that makes them so? Another way of looking at it is it costs energy to synthesize some exotic toxin. So an organism's not going to synthesize it unless it gets some evolutionary benefit out of it. So why do you think that they're so broad spectrum?
Because there's so many different kinds of zooplankton.
Yeah, exactly. There's lots of different kinds of zooplankton, and remember they're ancient over evolutionary time, so there could be lots of different iterations of it. Essentially, it makes sense to just evolve compounds that are toxic at a very fundamental biochemical level that all organisms have to operate on. And so it's no surprise then that those compounds are also toxic to people, to livestock, to dogs, and any other animal that drinks that toxic bloom. So this is one of the big issues. There's really not, on water quality, there's really not limits on the amount of cyanobacterial toxins that can be in drinking water in the United States right now. Australia, Canada, Europe do have limits. It's one of the few places we're actually lagging behind in regulating water quality. That said, if there are toxic blooms, most states very rapidly closed lakes to contact recreation and shut down the drinking water coming out of it. So it's fairly common. There's also some products on the market that are health foods called super blue-green algae, and they're basically concentrates blue-green algae. Most of it comes in around the health foods in the United States comes from the Klamath Lake. Those are probably not good things to take because those algae are known to produce toxins at times, and they can be toxic or not, and that's just the toxins we know about. The other thing that's going on is that scientists are repeatedly finding new toxic compounds, because there's that whole diversity of cyanobacteria out there, right? And then there's been other ones that have come up repeatedly. Other algae make toxins. So for example, this Premnesium is responsible for fish kills in Colorado. We've also had some in Kansas. And there are also important problems in fish culture ponds. Essentially, fish culture is this balancing act. You want to fertilize to get as much productivity out of the pond as possible, but you don't want to push it into a toxic bloom situation or the oxygen disappearing. So this is this push and pull between those kinds of things. So every summer we have fish blooms, fish kills. So this one is from this last summer. Kansas had 12 lakes with harmful algal blooms. Here's another one on USGS report on toxic blooms in Kansas. And I was just contacted by a woman from who just got her story on ABC, and she talked to people in Florida, Kansas, Iowa. All the states are having these problems. And it's not, there's not a reporting system that's set up for it, so it's really hard to get exact numbers on how widespread the problem is, because they're not required to report these as they would be, say, if there's a disease outbreak or something like that. Because cyanobacteria do better at higher temperatures than most other organisms, and humans are increasing the rates of nutrient input into aquatic ecosystems. Those two, and this is, both of these are happening globally, that we're having global increases in temperature on average. We're having global increases in nutrients because we have to feed more and more people, more and more population, right? And that part of that nutrient runs off into fresh waters. Because of those two things, cyanobacterial blooms and toxic cyanobacterial blooms are very likely to become even worse problems for humanity in the future. So it's not something that's going to go away, and it makes control of nutrient control even more important, given that the other conditions are setting up that favor these formation of these toxic blooms.
When you do get fish kills, that's not the toxicity, right? Is that just because it's coating the surface and it's making like hypoxic conditions, or how does that work?
Yeah, so it could be toxins, and it could be what you said, that using oxygen. So it's quite common that what will happen is that you'll get fish blooms just because you have an algal bloom of any kind that's not toxic. Those start dying off, or you have a bunch of cloudy days, so they respire more than they photosynthesize, and they use up the oxygen. Those conditions also tend to occur during summer when it's hot. And remember, when oxygen dissolves in water, well, I actually don't remember, I'll tell you this, but I sort of alluded to it. At higher temperatures, right, less gas can dissolve in water as opposed to salt, which increases the ability to dissolve in water. So at higher temperatures, there's less oxygen in the water already. Then you get this extra demand from the algae, and that kills the fish. So there's another area. That said, these hepatotoxins do work on fish, and that if you get trout or other species of fish that are exposed to a lot of this, it can also cause death to them as well. That Premnesium bloom that I talked about, that's a toxic effect. What happens is they bloom, they fall down to the bottom, and the catfish and the fish that eat stuff off the bottom just accidentally ingest a bunch of it, and it just tears them up inside basically and kills them that way. So sometimes yes, sometimes it's other causes. And you could imagine there being a combined effect, right, if you have a big bloom. One of the things that's, I think, worth talking about here in an ecological sense is when you expect organisms to make toxins, because there's all kinds of algae out there that aren't toxic or aren't very toxic, but usually they occur in mixed assemblages. Whereas the toxic cyanobacteria, when you find them, there's only one or two species blooming, right? And so you spread the cost, the risk and the cost of making that toxin. So we'll think about, for example, and we'll talk about this when we talk about resistance to grazing. again, but I'm gonna bring it up now so it gets you thinking about it. It's like hot chilies, right? You can't eat hot chilies as your only food source, and that chili is actually a defense, a plant defense. But if you have hot chili every once in a while in your food, it's not so bad. It's it's pretty good actually, right? So if every 100th algal cell that you're eating is toxic, it's not going to have much effect on you. Therefore, if you're an organism that's living in a bloom where everyone is toxic, then you're all protected. If you're living in a group of algae where most of them aren't toxic, you're essentially going to get selected against because it costs energy to build that toxin in the environment. So that's another reason, cyanobacteria, extremely dominant because of their ability to fix nitrogen, because of their gas vesicles. And then you add on to that, once they get dominant, then they can protect themselves from predators, and it all sort of feeds back on itself.
When you get a bloom, is it usually just one species, or is it a bunch of species blooming at once?
Usually it's one or two species. Sometimes it's a few more, but oftentimes it's pretty few species. So when even the bloom we saw in the Kimball Pond was one or two species for the most part, and the same thing with the ones in Milford and Tuttle Creek that we've had here over the last couple of years close to lakes. The most common hepatotoxins, microcystin. There's a number of other species that cause neurotoxic effects. But microcystis creates the compound microcystin, and that's the one that is easily assayed for. That's the one that they look for straight out as that hepatotoxin. The guy here on campus, Vandermeer, is he gets all the samples in when livestock or dogs die, and they've had a busy last couple of years. Last year was even worse than this year because not only was it warm, it was also calm, and so those blooms don't mix down, right? They stay up near the top. And that's when we had Milford closed, and we had the Tuttle Ponds closed, ponds closed, Tuttle, and all that. Right. Questions? Anything else? Okay, so now we'll move into the eukaryotes. As far as the eukaryotic algae, I'd say this is about the level that you should know things when it comes to testing. And mainly so you're prepping yourself for being able to do the ID in the lab. So most of this you're going to learn in the lab as you go through the samples. And you'll be required to be able to recognize these major groups. We'll just go through them. The chrysophytes, they're interesting because they're partially one of the reasons that you wonder about the idea between plants and animals, because they're flagellated, so they swim around and they ingest particles, so they can be heterotrophic, yet they're also photosynthetic. Probably most of this using particles like eating bacteria is to get nutrient, not carbon, but still they can use the carbon they get from that. The diatoms are an extraordinarily important group in both freshwaters and marine. Probably responsible for about 1/4 of the primary production on Earth. And their real key characteristic is this silicon frustule, this glass cell wall that they've got. And it's got very detailed ornamentation, and this ornamentation can be used to identify the species. It also breaks down very slowly, so it makes for, we'll talk about this in the silicon cycle, it makes for long-term changes in silicon in waters, but also they can be preserved quite well. So paleolimnologists, people that want to find out what happened over history in lakes, say over hundreds, thousands, or more of years, will take a course and they'll count the diatoms at depth and they'll date them with isotopes. They're used as well in forensics. You can, so if you drown, one of the last things you do is take a breath of water. I mean, you try to hold your breath as long as you can, but then you pass out and your body automatically breathes in, and the water rushes into your lungs, your alveoli burst, your heart's still beating, and so it takes the blood through your system for a minute or two until you're gone. But it gets all the way in your system, and so what they can do is take a a bone and pull the marrow out of it and boil it down in sulfuric acid to get rid of everything else and see if there's diatoms inside of there.
Yeah.
And so they've used this to show that, one, if people did die by drowning, so sometimes people get killed and they get thrown in the water, so somebody's trying to, show that they weren't, make it seem like an accident, right? Another type of case that's happened is somebody's drowned in one place and then they've taken them and thrown them somewhere else. They didn't want them to find out they drowned when they did, and you can use the local diatom assemblages that are in their body to figure out where they came from, if it was in a river or pond or something like that. And it's even useful because if you just find a body part, it can work. You don't have to have the whole body. So as long as you've got something that inner tissue that the blood had to flow into, and the diatoms wouldn't have just got in there by chance, that could also be useful. Dinoflagellates, they're also flagellates. Some are quite toxic, so some of the really nasty toxins, particularly in the estuarine system, are caused by dinoflagellate species, but there's also some freshwater ones that cause problems upon time. We saw the euglenophytes, so these are interesting organisms. They're light green algae that they have chlorophyll A and chlorophyll B. But they're unusual in that they have this flexible cell wall, and so they're capable of amoeboid motion and of using their flagella. Some of these groups are colorless; they don't have chloroplasts. Others do, and so they're this mixed group of essentially heterotrophs and autotrophs. And they're also, along with the dinoflagellates, mesokaryotic in that they oftentimes have an extra chromosome outside of the nucleus, so they're very strange organisms. And then as we get closer, the predecessors to green plants, the green algae and the charophytes. The charophytes become closer to the land plants because they start encasing their gametangents in sterile cells to protect it. And so that was part of the step that was required to get plants out of the water onto the land and keep their gametangent from drying out. And we'll see Kara, we'll collect it tomorrow at Edler Spring. We'll send somebody up there to grab it at least. I think there was some in pot 2. Was there some in the littoral zone of pot 2? Okay, so I'm just going to go through these groups. We already saw this as the cyanobacteria. And to give you an idea of some of the varieties, this is a chrysophyte. This is a common one, Dinobrion. I said it's flagellated. Right, and this is one we see in lake. It's quite large, 300 or 400 microns large, and these cells all sort of stick together. What do you think the adaptive benefit to being so large is? Yeah, that might be a little bit, although remember, larger objects have a lower surface area to volume ratio in general. But because they have this dendritic shape, then that probably, they're just bigger.
Doesn't sink.
Doesn't sink so much, but then also they can swim, so that's not as big of a problem. And then there's one other thing that we've talked about related to that.
They don't get ingested.
Yeah, there's zooplankton can't deal with a giant thing that's bigger than their mouth. That's a red alga, and we have these different sort of less common groups here. This gives you an idea of some of the variation in diatoms. Diatoms can either be centric, that is round, or sometimes barrel shaped, like the melosara that we talked about in the sinking and Stokes law, or they can be penning, and they can have a variety of different morphologies. And each of these are their frustules, and you count the number of lines per 10 microns and things like that to get the species ID. This one's kind of interesting because it actually has a cyanobacteria that lives inside of it that fixes nitrogen, so it's got an interesting symbiotic relationship. And we saw these, Ficus and Euglena and the Kimball pond. Here are some kind of flagellates here. This one also is fairly large and it's got these big spines, probably to protect it from grazing. And so this one and Dinobrian are two you'll often see in lakes with large zooplankton populations. And then this Chara right here, as you can see, it's getting more complex, looking more like it has stems and leaves, and closer to the progenitor to green plants. And these are the some of the green algae, just to give you an idea of the variation. And we go from very simple ones, a single cell with two flagella, to fairly complex ones that are almost like Chara with branching, right? And these are planktonic, these are benthic, planktonic. Spirogyra, maybe you've seen that one. Volvox, common in the intro to biology labs. Pretty neat that such a big thing can coordinate all its flagella and move around. And we'll see all these, a number of these in the labs. And this is another group of the green algae, desmids, again, to give you an idea of just some of the variety of shapes and forms that we